All right, in chapter 9, we're going to be looking at a few things about the cell cycle and then cell reproduction, at least one type of cell reproduction known as mitosis. Let's just start by looking at the cell cycle here in chapter 9, section 1. The cell cycle is what it sounds like. It's a cycle that our cells go through that includes kind of a growth type phase and a reproductive type phase. And we're going to talk about these two stages or phases here. You kind of see these at the bottom: interphase and mitotic stage or mitosis. We'll talk about a lot of details for each one of those. But it's a series of events or series of stages as that cell is living and then as it begins to go through its division. And so during this interphase stage, some of the main things are listed here that takes place. This takes place right before the cell divides, and so things are going on like the cell is growing, it's getting larger. Inside of the cell is changing because it's preparing for division, and so that means the organelles are doubling. You know, the cell is making new organelles that's going to go into a new cell when it divides. The DNA has to be replicated. The cell is going to have to make more DNA that's going to go into that new cell. So a lot of different things are going to take place in this interphase that we'll look at prior to the actual division of the cell, which is going to be mitosis. This chapter has some very nice figures, so make sure you spend some time looking at some of the different figures here. This is just a nice figure that summarizes the cell cycle process. And so it is a cycle. You could start really at any location to look at because it repeats itself, but a lot of times you start looking at interphase, and you'll see that there's really three main sections of what's called interphase here. There's two of which are growth phases, and so that's represented here by G1 and then G2. And then the S here, that stands for synthesis. That's where the cell is again growing in size, and it's replicating its DNA. It's replicating different organelles inside the cell. And then the M here stands for mitosis, or the mitotic stage or phase here, and they've listed out the different stages. Of course, you're going right here. This would be the first part, prophase, and then what's called prometaphase. So you kind of early form of the next stage is listed there, prometaphase. You have metaphase here, anaphase, telophase, and then cytokinesis is where the actual cell does its physical division and actually separates into two separate cells. So notice it is a cycle, and it does continue going around. And also notice that you can see from this diagram or this figure is that the length of time that a cell spends in interphase as a whole is much greater. So that's why they have this interphase here kind of going all the way around. The cell is going to spend much more time in interphase than it does in mitosis. So again, kind of refer back to this cell cycle from your textbook as we go through the notes. Interphase, just a couple of main points again, and a lot of this is on that figure we just looked at. So interphase, most of the cell cycle is spent there. So the time there, if you look at a microscope image, and we'll look at some lab simulations looking at mitosis. If you saw a microscope image of a cell, like a collection of cells, there's a good chance that most of them are going to be somewhere in the interphase stage, and fewer will be in the mitosis stage. And so the cell during this time is performing its usual functions. It's doing its living type functions. And so it's growing, it's developing, it's eventually going to start making new organelles, replicating its DNA. Not all cell cycles have the same exact time sequence, or at least time amount. In other words, time spent in interphase may be different from each cell type that you look at. Not all cells are going to have the exact same amount of time, but in general, you'll find the interphase lasts longer. And then we're really not going to get into really talking about nerve and muscle cells a lot, but there's a little distinction here between an animal's nerve cells and muscle cells, that they have a little different type of an interphase stage there. They really don't quite go through the same exact interphase stages as other cells do. So let's look at these main stages of interphase, G1, S, and G2. Now I know there's a ton of information here on this particular slide. We'll kind of go through it each one. But what I'm wanting you to kind of realize is some of the main takeaways about the cell cycle here, especially for interphase. G1, again, you can kind of think of this as a growth type stage. All right, growth stage. And it says up here, you know, the cell is going to spend 90% of its time roughly in interphase. Okay. And so about you've got G1. This is the first of the growth phases. Cell is basically recovering from just going through cell division. And so now it's growing in size. It's, you know, finishing up any kind of development for its cellular organelles that needs to be done. It's accumulating materials because it's getting ready to go through this whole process of division again. And so it's going to have to begin by going through DNA synthesis. It's going to have to replicate its DNA inside the cell to get ready to divide again. That division is going to take place during the S stage of interphase. And so this is where the main thing here is the DNA is synthesized, replicated inside, you know, during this time frame. There are some proteins that are needed, and we'll talk about that when we look at chromosomes. There are some proteins that are associated with chromosomes, and so they're going to have to be, you know, formulated and established during this time as well. I'll show you some pictures in a little bit of a chromatid, but chromosomes enter this S phase. They only have a single chromatid at that point, but during S phase, they're going to double their DNA content, so they'll have two chromatids. And I'll show you the picture of what that looks like in just a minute. But they'll double their DNA content, and so at the end of S phase, they'll now have two identical chromatids, which are referred to as sister chromatids. Those chromatids are going to be attached, and that kind of gives you, if you've ever seen a kind of a drawing or a schematic of a chromosome, it almost looks like a letter X, but that kind of gives you that X-shaped type chromosome that maybe many of us are familiar with. Those are made up of two sister chromatids that remain attached until they go through the mitosis part of cell division, and they will separate there. Last stage of interphase is another growth type phase. This is right after the synthesis, the S stage, and right before the cell goes through mitosis. And so here the cell's kind of finishing up any preparations, getting ready for division, making some more proteins. It's going to need some microtubules. These are going to be some proteins needed to kind of move the chromosomes around inside of the cell during the mitosis stage. Yeah, so here's the picture. This is figure 9.2, and it's kind of a nice little figure. It's very simple though. Here's a chromosome, which is made out of DNA strands and protein. We'll talk about that. But it only has a single chromatid. This would be a single chromatid here. During the S stage of interphase, that DNA is going to replicate, and so you still have a single chromosome here on the right side, but now you have double the amount of DNA. So now you have two chromatids. They're identical. They should be identical. You have two chromatids, two identical sections of DNA here. They are joined somewhere. It doesn't always have to be in the middle. Sometimes they're joined at the end, but where they're joined, this is known as a centromere. And so now you have a duplicated chromosome, still a single chromosome. Then once it goes through cell division, once it goes through the mitosis, during the stages of mitosis, we're going to find out that this chromosome is now going to divide and will be reduced back down to a single chromatid because that cell is going to split. And so one chromatid will go into one cell, one daughter cell. The other chromatid goes into the second daughter cell. All right, so let's start talking. So that's a little brief introduction to interphase. G1, the S, and G2. Now let's look at mitosis, or the mitotic stage, the M stage. It includes a couple of things. One is the mitosis, of course, and that's the actual nuclear division That's where the cell is dividing itself, dividing up what's going on in the nucleus, and that's going to be looking at the chromosomes. That's where we're talking about prophase, metaphase, anaphase, telophase, what's taking place in the actual, what's taking place with the DNA in the chromosomes. Cytokinesis, this is literally saying that this cell is going through a complete division now. It's the division of the cytoplasm. This is where you actually separate the cell into two, hopefully identical daughter cells. They should be identical, and I kind of mentioned that, unless something happens, and later in the chapter it kind of mentions something like a cancer type scenario where the cell divides kind of uncontrollably. And so, or maybe there's some kind of mutation that takes place in the cell. And so, but as everything goes correctly, this mitosis should result in two genetically identical daughter cells. So there are some controls of the cell cycle in place that should control things, so that things, the cell cycle does go in a sequence of events, that it is have a certain amount of time frame that it spends in each cycle. And so there are certain signals that work to help influence what's going on in that cell. And some of these signals are external. Some of these signals are internal to the cell. An external signal is something that's coming, some type of protein signal that's coming from usually like what's called a growth factor. It could be like a hormone. It's a signal that it will attach, that maybe go through the bloodstream. It'll attach to the plasma membrane of the cell, and that triggers some type of internal control mechanism inside the cell. But it's an external signal. It's coming from outside the cell, initially initiated outside the cell. An internal signal is some type of protein that's already inside the cell. This is where these are called cyclins, which is kind of a nice name because they're controlling the cell cycle. They're controlling the timing of the cell cycle, making sure that certain checkpoints are met. The G1, mitosis, the S phase, the G2, there's certain checkpoints that need to be met at each stage to make sure that everything is in time, everything's turning on like it's supposed to, everything's turning off like it's supposed to, so that things don't duplicate faster than they need to, or out of control like some type of tumor cell might. And that's why it says down here that things like solar radiation, UV light, or higher energy sources like X-rays can cause problems with those cyclins that interrupt this G2 checkpoint, so that the cell doesn't know how to go into the mitosis stage correctly, or that it may go a little too fast, and so now the cell is dividing at a faster rate than it really should. Another part of cell cycle control is what's called cell death, or programmed cell death. This is a good vocabulary word. You'll probably see a question about this on the next test, but apoptosis, kind of a strange word, apoptosis. This is programmed cell death. And so there's certain cell events that take place that basically tell this cell when it's time to kind of finish its life cycle. And so some of the things that take place is the nucleus literally starts to break down, the plasma membrane starts to break down, and eventually you have white blood cells, some of your immune cells, that will come around and kind of break up and engulf and kind of clean up those cell fragments. Some of these events, like breaking down the nucleus and breaking down the plasma, are caused by certain types of enzymes, digestive type enzymes. These are called caspases. And these are kind of breaking down and trying to degrade the cell organelles inside and break down the cell. And so cell division and cell death, apoptosis, occur throughout our lifetime. So cells are always going through these types of things. You're always making new cells, but you're also getting rid of some old cells as well. And so it says that an abnormal cell, sometimes there could be an abnormal cell that doesn't get the signal, or maybe something's wrong with its cell cycle. It may become cancerous. Sometimes you have maybe a virus infected cell that needs to be, broken down. And so apoptosis could hopefully get rid of these types of scenarios. So apoptosis, programmed cell death, could get rid of that virus infected cell or even a cancerous type cell. And so that's why inside of the cells, they have specialized vesicles, storage containers that are holding on to these enzymes that hopefully will, work when they need it. When they need these enzymes to work, they're available to, get that cell to basically digest itself. One signaling type protein here that's a lot of research has been done on is called P53. And so its job is to basically stop the cell cycle or halt the cell cycle if it detects that there's some kind of damage to the cell's DNA. And so it wants to stop that cell cycle at that G1 stage so it doesn't go any further. The idea is hopefully if you stop the cell cycle at that G1 stage, it can fix the problem, can fix the DNA repair, and if so, it can continue on with its cell cycle. If it can't fix it, or if the DNA doesn't repair itself, then the apoptosis, the programmed cell death, is initiated and you get rid of that cell. So it's kind of a little safety protocol there to try to fix the DNA if it can, or if not, get rid of the cell so it doesn't turn into some type of cancerous type cell. All right, section 2 kind of takes a closer look at what is the chromosome like, and really section 2 is looking at the eukaryotic chromosome. What is the chromosome like for eukaryotes? And so here's just some description things about it. One, it contains a single DNA molecule. We got the DNA double helix, and we're going to look at DNA a little closer as we get into some of the other chapters coming up. We're going to see the composition of DNA. We know already that it's a nucleic acid that it has, base, nitrogen bases. It's got a sugar phosphate backbone for the nucleic acid, but here we find that DNA is also associated with certain types of proteins. These proteins are called histone proteins. And so when you take the DNA, and again, there's another figure that we'll kind of look at in just a minute, but we take this DNA and we start to coil it up and wrap it up with these histone proteins, that's when all of this together is referred to as chromatin. And this is what's the structure for a chromosome. Now, a couple of things about the histone proteins. Most protein, or a lot of proteins, do have a structural type of a role, and that's what these proteins are for. They're kind of holding a structure for the chromosome. They do tend to have certain types of survival functions here. In other words, they're protecting the DNA. There's 5 main types of histone type proteins, and they're mainly responsible for packing the DNA, winding it up, compacting it, getting it smaller and tighter so that it will go inside of the nucleus. Now DNA winds itself around really a core of 8 main histone molecules, 8 units of proteins, histone proteins. And once that happens, it's referred to as the nucleosome. And it almost looks like a string of beads. And we kind of have a little picture there. The figure 9.4 tries to illustrate some of this. It's kind of small here, but at the smallest level, here you see just the DNA. That's the double helix. That's the repeating nucleic acid sequence. And then that DNA is wrapped around these histone proteins here. And that kind of looks like in multiple places, you start looking at these, and it looks like little beads on a string. And here's a electron microscope image that kind of illustrates that. It kind of looks like little beads on a string. That continues to kind of wrap and coil together. And that's when you start forming what's called the chromatin. Once you continue to wind this together and you make it more compacted, it compacts together, makes it more tightly coiled, that's when we have what we refer to as a chromosome. So a chromosome is DNA, and it is these proteins, but it's all tightly wound together to form this chromosome structure.
All right, in section 3, we'll look at a little closer to the stages of mitosis, and eventually talk about the end result, the cytokinesis part of it. A couple of things to think about here. And again, we're looking at eukaryotic cells and the process of cell division. One thing to realize is that mitosis really includes both a nuclear type of division and also the division of the cytoplasm. And so anytime you see it referring to nuclear division, we're talking about the actual division of the nucleus inside of the cell. And then at the end of mitosis, we'll see where the actual cell divides, a cytokinesis, which literally means division of the cytoplasm. And so that's when you would have two daughter cells resulting from that single cell that's going through mitosis. We've already mentioned a little bit about sister chromatids, and we'll show you the picture again of a duplicated chromosome. But we're going to see how in the stages of mitosis, the sister chromatids really become separated and kind of move around in the within the cell, and then eventually will be distributed into individual daughter cells. And so a couple of things that happen right before mitosis begins is that the chromatin, remember chromatin is your DNA kind of wrapped around the histone type proteins. That chromatin condenses, it essentially coils up very tightly. And so now at this point, you would actually be able to visibly see chromosomes using a very simple light microscope. You'd be able to see those chromosomes because they're now very shortened, they're thicker, and they're more dense at this point. And the other thing to kind of think about is each species has a characteristic chromosome number. So humans have a chromosome number. We'll talk about that. You have a diploid number of 46 chromosomes, or a haploid number of 23. I'll show you that coming up. But if you looked at a plant or a type of bacteria or other types of animals, you would find out that different animals and different plants have different numbers of chromosomes. So again, here's the picture, and I'll refer back to the picture here of a duplicated chromosome. And we've kind of mentioned a few things about it. Remember, each one of these are chromatids. So this is after the S phase would take place in interphase, and so the DNA has now replicated or duplicated, and now you have this shortened, thickened type of a chromosome here. Each of these are sister chromatids because they should be identical copies of each other. They are joined in a location known as the centromere. Now this particular one shows the centromere almost to the center here, but it doesn't always have to be right in the middle. It could be a little further up at one end or the other. The other thing is just outside of the nucleus is another organelle known as the centrosome. And this little figure down here kind of shows, you can kind of see the cell. Here's the nucleus. You've got the nucleolus inside. You've got the centrosome located here. This is going to help organize a series of microtubules. Remember, when we talked about microtubules as being tube-shaped protein type structures, long fibers of protein. They're in a kind of coiled up into a tube type structure. These microtubules are going to form what's known as a mitotic spindle. The spindle includes many different protein fibers that will essentially attach to the centromere area at a specific area known as the kinetochore. Right, so we've got our sister chromatids. They're joined somewhere along that chromosome in a centromere location. Then you have some protein that's located here on either side. For each sister chromatid, you'd have a kinetochore. This is where these spindle fibers would attach. And again, eventually we're going to talk about the different parts of or stages of mitosis: prophase, metaphase, anaphase, telophase. These are where now that you attach a spindle fiber to this kinetochore, you can actually the chromosomes actually will move around in the cell and eventually separate the sister chromatids, because the idea is, you want to separate this chromatid to send one chromatid into a new cell, a new daughter cell, and the other chromatid will also go into a different daughter cell. So let's look at some of these main stages of mitosis. And so prophase is the first stage here. Some of the main characteristics about prophase. Nuclear division is taking place, is about to take place here. In other words, the nucleus itself is starting to essentially break down. And things that are happening in prophase, you've got your chromatin that has become condensed. It's coiled up, it's gotten thicker, a little shorter. So now you can visibly see chromosomes if you have just an ordinary light microscope. The nucleolus or nucleolus disappears from inside of the nucleus. That nuclear membrane around the nucleus, the nuclear envelope, it starts to break up. In other words, all of the DNA, of course, is in the nucleus, but now you need to get that DNA out of the nucleus, out into the cytoplasm, so that you can allow for cell division to take place. So early prophase is all about kind of getting those that chromosome formed and getting it into the cytoplasm. So that's why the nucleus starts to kind of, the nuclear envelope starts to break up a little bit. The next main phase is metaphase, but sometimes you see certain textbooks kind of have an early form of metaphase or early stage of metaphase, and so they'll refer to this as prometaphase. It's just before the full stage of metaphase takes place. And so here you've got preparations for starting to get the sister chromatids to start to separate. And so, for example, that centromere that I showed you, that section where those chromatids are connected, they develop the kinetochores. Again, the kinetochores are made out of protein. They're attached to the sister chromatids, and they're going to be binding sites for the spindle fibers coming out from the microtubules, and they're going to be binding sites so that you have an attachment there of these protein fibers to allow for movement of the chromatids within a cell. So let's look at metaphase next, fully now in metaphase. I like to think of meta here as being middle, because that's kind of what's happening here. The chromosomes are being essentially pulled by these fibers, and they're going to essentially the middle of the cell. Chromosomes are lining up. I'll show you some figures and diagrams in just a minute. But the chromosomes, if you can kind of imagine, the chromosomes are going to the middle section of the cell. And so technically that area is known as the equatorial plane, the metaphase plate. You'll see that the plane right in the middle where these chromosomes are lining up. You've got the spindle fibers or the kinetochore fibers attached to those kinetochore protein areas, and they're basically making sure that your chromosomes line up right in the middle of the cell. In anaphase, now the centromere that was kind of holding the two sister chromatids together, it starts breaking down, and that allows the sister chromatids to start to physically separate. They're being pulled apart to go to the two polar regions of the cell. Eventually, the last stage here is telophase. At this point, there's no need for the spindle type apparatus. The mitotic spindle now is going to start to break down. You start forming new nuclear membranes or nuclear envelopes that's going to form inside of the cell. It's almost like you're kind of putting the cell back together at this point, so that eventually, and we're going to get to the cytokinesis phase, eventually you're going to divide the cell in half so that you'll hopefully have two identical daughter cells. So things that have to kind of be reversed. You're building up the nucleus once again. You've got your chromosomes are still kind of separated now into, they've moved apart, separated, and you're putting two new nuclei, you're forming two new nuclei around those daughter chromosomes. And then you start the process of cytokinesis, which is going to be physically dividing that cytoplasm in half. The other thing that's taking place, again, we're trying to kind of reset back up everything. Your chromosomes that were once really thick and condensed and coiled up, tightly coiled, they're going to start to diffuse out again. They're going to start to get smaller and kind of stretched out and less condensed again. And so, in other words, they're becoming chromatin again, just like you started with at the beginning. So when you kind of look at some of the diagrams, I know it's kind of hard to see here in the video, but go back and look at figure 9.6. It does a good job what they do is they show you an example of a cell, just a general cell here. They show you, they kind of follow through an animal cell, a typical animal cell. They follow through a typical plant cell on the bottom, and then they highlight some of the main key points, key takeaway points from each stage. So prophase, early on, you've duplicated the organelles, you've duplicated the centrosomes. Your chromatin is condensing down into chromosomes, so you can start seeing them appear visibly with a microscope. The membrane around the nucleus is starting to break down. And then, eventually in late prophase or during the actual stage of prophase, now notice in this cell, you no longer see the nucleus. You see the chromosomes that are here. These are duplicated chromosomes. They've got two, chromatids attached at the centromere. The spindle fibers from these centrioles have now started forming, and they're going to branch out and eventually attach to those kinetochores of those chromatids. Metaphase is starting here, the prometaphase area, and as you're starting to attach those spindle fibers to the kinetochore area. Metaphase proper here, metaphase, you're going to have the chromosomes line up in the middle of the cell. Notice in this whole example here, I didn't mention it earlier, but this whole example here, you're dealing with a cell that has 4 chromosomes, 1, 2, 3, 4, and they're all lining up now in the middle section of this cell. Anaphase is when the individual chromatids, sister chromatids, now begin to separate. That centromere has now essentially dissolved or kind of broken down. And so now you get the spindle fibers that are direct and are kind of pulling these chromatids to separate poles of the cell. Also notice here that the cell is starting to show some signs of cytokinesis. It's starting to indent a little bit right at this mid plate area of the cell. And then in telophase, you do a couple of things. One is, again, you're starting to make this indentation a little bit more, so it's a little more clear that you're getting ready to go through cytokinesis and divide the cytoplasm right down the middle here. You're also starting to rebuild the nucleus on both poles of this cell. Technically, this is still a single cell that you're looking at, but here you're starting to, all of the chromosomes are now centrally located, and they have this nucleus that's starting to reappear and reform around the chromosomes. Also note here that I still have 4 chromosomes. They're just half the DNA content, right? So they're not duplicated anymore. I don't have sister chromatids anymore. I have 4 individual chromosomes here, and then I've got 4 individual chromosomes in this other cell that will eventually be, 2 daughter cells. So at the end of this, at the end of telophase, you have cytokinesis, and again, that just means division of the cytoplasm, division of that entire cell. And so that means that, half of the original cytoplasm and half of the DNA is now going to be distributed equally to two daughter cells. And we saw where a little bit of this process actually started taking place as early as anaphase with a little bit of that indentation there. Now, one thing that kind of stands out about animal cells is you have what's called the cleavage furrow. And this is just kind of a little division area, kind of right here in the middle. This little, kind of a little light line is kind of hard to see here, but this is the cleavage furrow here, this indentation that here is where that is going to take place. You really don't see any kind of cleavage furrow that appears in the plant cells, and that's because you've got the cell wall here. You do generally see a line here that does develop, but not really like a cleavage furrow like you would see in an animal cell. For plant cells, you generally see what's called a cell plate. The cell plate forms. It's more, again, that's because of the plants have the cell walls, and so you have more of a structured cell wall that's going to be dividing right in the middle rather than a furrow that's evident in an animal cell. And that's kind of what this is showing. This is just kind of showing the cytokinesis part of this. Cytokinesis is taking place early, starting in anaphase, continuing in telophase, and then finishing at the end of telophase. You would have the separation of the cytoplasm eventually. When you think of mitosis, why are cells dividing? This section kind of mentioned a few things, of course. Obviously, cells have to grow, but they also have to repair themselves. They have to divide to increase not just their numbers, but also to replace cells or older cells or broken down cells. And so they need to have repair mechanisms as well. And so this is just kind of talking some examples of growth and development and repair here. We're talking about a plant, a flowering plant. You've got certain types of tissues that essentially are like stem cells, and they call these meristematic tissues. They have the ability to divide throughout the life of the plant, and so they can continue to go through cell division and mitosis. In animals, mitosis is needed. And they kind of give you some examples here. You have a fertilized egg cell. It's going to start to grow into an embryo that's going to eventually become a child, a baby. And so, mitosis is where you have, the growth of the organism to make new cells. But it's also when you have cells that have to be repaired. And so, if you've ever had any kind of cut or broken bone or damage to your tissues, then mitosis is the means by which you're going to replace those cells. Certain tissue types have more cell division than others. So, if you think of your skin tissue, that's epithelial cells. They go through mitosis at a much faster rate than bone cells would. Section 4 mentions a little bit about the cell cycle and cancer, and we'll just mention a few vocab words, talk about a few things here. Anytime cells grow out of control, you have some type of abnormal growth. That's called a tumor. Not all tumors necessarily are cancerous. Cancerous means that they actually begin to spread generally. And so some tumors are what are referred to as benign. They're not cancerous. They seem to have like a capsule of tissue around them. So they're isolated, and they usually do not travel or spread or invade neighboring tissue. Malignant, the prefix there, mal, means bad, and so malignant tumors are cancerous. These are not generally encapsulated, so they're not enclosed, so that means they have a little easier ability to spread into neighboring tissues. They may also detach from the location where they're growing from and perhaps even spread to distant places. You may have heard of metastasis. And so this is just where cancer spreads. Sometimes it makes its way to the bloodstream or the lymphatic system, and that causes it or allows it to spread to different locations in the body. In general, you can think of cancer cells as having certain types of characteristics. They lack differentiation, so that means they're not specialized. What that means is a cancer cell may develop in a particular organ that may have, if it's the liver cells, then those are hepatocytes, but they may the liver cells may start to grow abnormally, but they no longer function as liver cells. So they don't really become liver cells anymore. They're just non-specialized cells. They can be essentially referred to as immortal. In other words, they don't have the same types of controls that other cells do if you think back to the cell cycle. And so they just continue to repeat the cell cycle without any type of control. Many times you'll see cancer cells having nuclei that are perhaps enlarged. Maybe they have the incorrect number of chromosomes. Maybe they have an extra copy of a particular gene that shouldn't be there. So sometimes something is wrong inside of the nucleus. And generally, cancer cells don't go through that programmed cell death process we talked about, apoptosis, like they should. And so again, something along the way, some type of signal has corrupted their programming, if you want to think of it that way, so that they no longer get that signal to actually go through cell death or apoptosis to try to control that population of cells. There's a nice table here, table 9.2. It just kind of gives you like a little breakdown, easy way to kind of organize cancer cell versus a normal or non-cancerous type cell. All right, section 5 we'll look at briefly. We've been talking about eukaryotic cell division. Prokaryotes, our bacteria, they are going to go through a type of cell division also. There are some differences we need to kind of address here because the structure of the cell is different. We still have DNA there, but remember the prokaryotes do not have a nucleus, a defined nucleus with a membrane around it. And so the DNA tends to concentrate, though, in the cytoplasm in an area called the nucleoid. The nucleoid just means it's like a nucleus, but it's not. It doesn't have the same type of plasma and membrane wrapped around it like a eukaryotic cell would. And generally, we talk about, for bacteria, something known as binary fission. That prefix bi means two, and fission is a division or split here. And so literally this is saying that bacteria have the ability to divide in two. And so they can replicate chromosomes. They still have chromosomes. Many times the bacteria chromosomes are in a circular type format rather than kind of that characteristic X-looking shape chromosome that we're used to for a eukaryotic cell. But you can see chromosomes, they have to replicate as well, and they're going to distribute out into two identical daughter cells. Essentially, this is an asexual type of reproduction. And you don't necessarily have to memorize this, but if you have ever heard of a bacteria known as E. coli, you generally find this in your intestines. It has a generation time of about 20 minutes. So within 20 minutes, that population of E. coli has ability to go through binary fission. So that's a very quick, fast replication. And a lot of bacteria have a very short generation time. They can replicate very, very quickly. And so there's a few steps here. You can kind of look at the diagrams. They do a good job of kind of showing some of the stages here. Here you have a bacterial cell. Notice that it does have a cell wall. Notice that the chromosome is not like a eukaryotic chromosome. You kind of have a circular type of a chromosome. And it's not really in a nucleus. It's in the nucleoid region. So that means it doesn't have a membrane around it. But that chromosome needs to divide as well. So that chromosome will duplicate itself or replicate itself. And now you have two identical, in this case, circular chromosomes. And then the cell will start to essentially go through cell division, binary fission, and divide into two daughter cells. Notice what was missing from eukaryotic cell division. Notice we didn't go through things like Prophase and Metaphase and Anaphase and Telophase and all the different stages. You didn't have the spindle fibers. That you don't need those spindle fibers attaching because you just have that main central chromosome that needed to duplicate and then to divide to go into different cells. It's binary fission.